Eukaryotic cells are normally much bigger than prokaryotic cells and contain a membrane-bound nucleus and other membrane-bound organelles, organized structures that perform specific functions. At the outer surface of an animal cell is the plasma membrane. This remarkably thin covering controls the movement of substances into and out of the cell. The plasma membrane is also involved in the recognition of one cell by another, in receiving hormonal and other signals, and in binding pairs or groups of cells together. Almost all of a eukaryotic cell's DNA is contained within a membrane-bound nucleus. Most of the genes in a cell's DNA contain information for the production of proteins. This information from a gene is copied into a messenger molecule that exits the nucleus and encounters a ribosome. Ribosomes are the sites where protein synthesis occurs, translating the messenger's code into protein. Ribosomes may be suspended free in the cytoplasm or attached to an extensive membranous network called the endoplasmic reticulum, or ER. Proteins are threaded into the lumen of the ER, which in this region is called the rough ER because it's studded with ribosomes, creating a rough texture. In the lumen of the rough ER, proteins are processed and modified, enclosed in vesicles, and transferred to the Golgi complex for further processing and packaging. Proteins made on the rough ER will be incorporated into membranes or secreted by the cell eventually. Proteins produced by free ribosomes will remain in the cell. A membranous network called the smooth endoplasmic reticulum lacks ribosomes. The smooth ER, particularly in liver cells, detoxifies alcohol and other drugs such as phenobarbital. Another function of the smooth ER is the production of additional membrane material. The Golgi complex consists of flattened membranous sacs. The Golgi complex receives proteins from the rough ER and then chemically modifies many of the proteins as they move, by way of vesicles, from one membranous disc in the sac to the next. The Golgi complex sorts the proteins and sends them to their various destinations. One destination is the plasma membrane, where the proteins are either exported from the cell or become part of the membrane. Some protein-filled vesicles become lysosomes. Lysosomes are packed with about 40 different digestive enzymes. The enzymes and membranes of lysosomes are made by the rough ER and processed in the Golgi complex. The lysosome plays numerous digestive roles in the cell, including digesting substances brought into the cell and destroying and dismantling obsolete parts of the cell itself. Mitochondria provide the cell with most of the energy it needs. The metabolic processes that extract energy from food molecules occur partly in the cytoplasm, but mostly in the mitochondria, and result in molecules of ATP, the cell's primary energy currency. The structure of a cell is determined in large part by a network of fibers that traverse the cytoplasm. These fibers make up the cytoskeleton, which provides the cell with support, movement, and a means for dividing a cell into two parts during cell division. The micrograph depicts a network of cytoskeletal fibers called microtubules.